Why is it no? No is one thing, but knowing why no is the second part. Bingo. Yes. 1 point for it. All right. So your heart muscle is a striated muscle because it has to have the convection power. But you do not have control over your heart. You're not sitting there and saying 64 heartbeats. 2, 3, 4, 5, 6, 7, 8. No, you don't do that. All right. Got that. When you grow older, you tend to turn white-haired, like me. Does this affect skin color as well? Do you bleach when you grow older? When I go in the sun, I turn even whiter, but my face turns red, and after that it turns brown again. So what does it tell us about certain cells you have in your skin? Like melanocytes. Will you lose your melanocytes over time when you age? Yes, no, maybe. What do you think? What is happening in your skin when you grow older? You see it getting more foldy, right? Which means there's a number of things that you lose. But the skin color is really not going away. Maybe slightly going down because you are less often exposed to the outside. The melanocytes stay with you. Okay? Your melanocytes remain. Does it matter what you eat? Or is every food just food? You can gobble it in, you go to fast food, or you prepare your meal at home. Doesn't really matter. It always has exactly the same thing, calories and everything else you need, but you need to be a little bit more choosy. And if you are a little bit more choosy, what are the things you really are looking for? What are the food content you're particularly interested in that is running under the quote, healthy food? Going to McDo and eating fries every day, is that healthy food? Good, yeah, we got that part. What would you eat instead when you go to McDo? You get yourself a salad. And they offer that now. What do you want to get out of salad? Vitamins, here we go. What else do you want to get out of your food? When you break it down, remember we go through all the stomach digestion. We take our proteins and chop them up into the amino acids, and then we run into certain problems. Not every food has every amino acid your body needs. The only way you can be absolutely sure is to drink mother milk all your time. Well, your mom would be really complaining about you coming and drinking mother milk now. Second part is you eat your neighbor and he will complain when you chop on him, right? So not every protein that is out there provided by nature has every amino acid you need. There's a bunch of amino acids which we call essential. So you need to think about that part. And last but not least, we talked about fat. You want to take your butter and put it on your bread. This is good butter. You know your mom told you eat good butter, but it's not good for you because the butter does not contain a number of fatty acids you need. We talked about Mediterranean diet, so there you have a little bit more along that line. Right? So, does all food contain everything and is just food? No. These are the components you're looking for. So what do we really get of our pancreas? Why do we have a pancreas? And we only talk about the exocrine part of the pancreas. Yay! Thank you, darling. I'm tired now. You took too much energy out of me. Why do you have a pancreas? So it fills your belly. You get a nice protrusion coming out of it? Nah, not really. What else do you want to get out of your pancreas? Enzymes. What kind of enzymes? They break food down. What kind of food are they breaking down? They want to have enzymes that are breaking down proteins into amino acids. And these produce a very low pH, pepsin. And we talked about the guys coming out of the pancreas, trypsin, chymotrypsin, and they require a alkalic pH. So we need to have something else produced out of our pancreas. Bicarbonate. Because your bicarbonate buffers against the acid coming out of the stomach. Okay? So this is really the two components you want to keep in the back of your mind. What is the exocrine function of the pancreas? Did you cover today in the lab the endocrine function as well? So, absorption of nutrients out of fat, protein, carbohydrates happens where? Right there in your mouth? Or do you have to cut some hanging on? Where's all of that good stuff you need to be sold? Right in your rectum? Shortly before you go to the restroom? Not a good place. Small intestine. All right. What helps coordinate the information flow between different heart muscle cells? We talked extensively about it at two different lectures. One lecture introduced the connection between different cells when we talked about tight junctions and stuff like that. When we put it in the context of how heart muscle is functioning, we have electric current spreading from cell to cell. What kind of junction do you need for cells for that to be able to do so? Anybody? Tight junctions is only epithelia where you literally have to seal off. Now you need to have the content of cells being in continuity. This to do with gap junctions. Yay, there we go. Okay? What are the layers of our integumentary system? And how many layers can be distinguished? I think you go when you go in the skin before you hit the bone, or the muscle actually before you hit the bone. What are the layers of your integument you will see? How many? 3, 4, 5? 5? Who is more than 5? Who is less than 5? 4? Was that a 4? A 3? Who is going for the 3? Anyone going for the 4? Anyone else for the 5? The 3 has it. Alright, so how we call these layers? Start out on the outside, epidermis, and then underneath, dermis, and underneath, hypodermis. There we go. Voila. Learning experience number 1. Get them all squared away. Alright. Jump a little bit into a different area. If we accept that genetic similarity indicates ancestral relationships, can this be used to argue about the origin of man? So remember, you go out, you are a biology student, and you want to figure out how closely are two flies related to each other. You basically take them, grind them up, look at their genes, display their genes, look how close the genes are, and then you can say, they are pretty close, that belongs to one species. They are slightly more separated, they are two different species. That's in a nutshell how we do it nowadays. So now we do the same thing with us and the great apes. And what do we find? We have a lot in common with our close living ancestors. So, if we accept the generalized approach that evolution is changing slowly but surely the DNA, and the more similarities you have, the closer you are related. How do we know this for a fact? What is the closest relationship you have genetically among the people on this planet? Any one of you is a monozygotic twin? Nobody? Okay. This would be your closest relationship. relationship. Because you would have the same genes. Nobody? Okay. This would be your closest relationship. Because you would have the same genes, right? The next one you have are your six, and your mom and your dad. Okay, and then it gets a little bit less. So if you follow that part, mom and dad had a grandfather, grandfather had a grandfather, and on and on, what you really do. That's how we can then, looking simply at the distance in terms of genetic diversity, and say, yeah, it was split about 1 million years ago. But one more point, I pointed out very extensively how we can really correlate that part is, we know exactly 2 chromosomes defined in big apes, chimpanzees, gorillas, and orangutans. They are fused together in the human to form a new chromosome. So we have 46, and all those guys have 48. Okay? So this is most in science considered to be a very good evidence, and I showed you all the details about it. So we talked about this already. What do you think? How many genes do we have in common with the chimp? I know we don't like the idea, but yes, what percentage? 98.8%. Anyone else? More or less? 50%? Anyone go 50%? 60%? 70%? 80%? 90%? 98.8% have it. All right? So it's very similar. Very similar. Over 98%, all right? So moving back to something else, what is the function of those guys? Why do you have bile? Yeah, not a good idea to think about. You have your gallbladder in you, and what are the lipases doing in your fat digestion? So you need to break down your fat in its constituent parts. So if you drink your mother's milk, it comes in lipid droplets. And what you need to do with your lipid droplets? You make it even smaller. We call it emulsification. All right? And who's doing that? Who makes the lipid droplets even smaller? Your bile. That's how you have it. Whenever you eat a food very rich in fat, there's a response to your gallbladder will be excluding its content. Your bile is mixing with the fat and breaks it down into even smaller components. Because only very small areas can be attacked by the lipases and cut into its constituent parts. You can't have a big oil droplet literally going through the digestion. You break it down into smaller parts. So your bile is the emulsifier, and your lipases are doing the digestion. All right? We talked about the problem of cancer only very short in the context of long-term homeostasis of cell replacement, and I mentioned the word metastasis. So what do you mean with that word? How does it work?
Spreading to the other parts of the body.
Spreading to the other part of the body. And how do you do that? Going and walking, slowly but surely move your way through the different cells. That is the proliferation of the same thing, but what we are now looking at is exactly what he's talking about, going from part A, where you started out as a cancer to proliferate, to part B, that is until now normal. And what you do is, you find a blood vessel, you hit your right, you take the blood's own circulation system to go throughout the body, and then you find a nice wide place where you can get out of the blood vessel and build a second cancer. Okay? So metastasis literally is the spreading of the cancer throughout your entire body. Okay?
What you need for it is essentially blood vessels. It's already there, it's a highway. It's like your I-80. You want to go from here to Des Moines, you're not driving over the cities, you're going on to I-80, right? Which one is the worst skin cancer? You don't want to have any cancer, I know that. But if you would have a choice and somebody would say, no, here are the 3 different kinds of skin cancer you can have, which one would you choose? Or not choose, to be absolutely sure about it. Melanoma, there we go. Okay? Staying within the cancer field, so what do you think is the highest incidence of new cancer per year? Breast cancer, pancreas, prostate, colorectal cancer. Skin cancer. Skin cancer, the new cases per year, 1.5 million. Skin cancer alone is about 1 billion. Okay? It's very epidemic right now. Everyone done on that part? Now that is now a dead giveaway because we just went through the entire thing. Sudden changes in blood sugar, blood pressure, blood pH, blood temperature cause always what? It activates a regulation system. How do we call that regulation system? A feedback loop. How do we call that feedback loop? A negative feedback loop. Okay. So you literally have a set point for all of that, and the negative feedback loop is regulating to go back to that set point. Precisely what you just heard about 20, already half an hour ago. Okay, it's lost already in the short-term memory. All right. But that's really the basic principle. Your entire body homeostasis is always following the same principle, with very, very few exceptions where you have a positive feedback loop, like if you go into labor, which would only hit about half of the class. Right? Because guys don't typically give birth. Okay. Are all substances produced in accessory glands of the digestive system secreted into the lumen? That's a trick question. So we talked already about the pancreas. We talked already what you get out of the pancreas. You want to get all your digestive enzymes coming out, and you want to get bicarbonate coming out to neutralize the acid from your stomach. Right? The pancreas is doing something else. Is it producing something else? Is it only what we call an exocrine gland? What would happen if I take out your pancreas? Actually, I go in and take out precisely only a subset of cells, called the beta cells. Because the alpha cells are the big guys who secrete all the rest, or we're looking at the beta cells. If I remove all of that, you would not live very long because you would have a problem in regulating your blood sugar. Are we now clear of what we're talking about? Blood sugar regulation is done by hormones. What is the hormone we are talking about? Insulin, right? So your insulin is produced in your pancreas. Okay, so there are hormones that are also produced that is not secreted out in your guts. Why would you want to do that? If you secrete your insulin in your guts, what would happen to the insulin? It gets digested. There's no effect. So we need to put it someplace else. Okay? The place else in this case is blood, because that's where we need it for the blood sugar regulation. Is that clear? Everyone with me? So we talked already about peptidases. Where do we get them from? Pancreas is one. Where else do you get peptidases from? What do we do in the stomach? Which one is that? Pepsin. And for pepsin, we need to have for its function a very low pH. So now we bring it all together. All right? So you have at least two. pepsin from the stomach, and trypsin from the pancreas. That's at least a good start for your digestive system. A little bit on the genetics. Okay. So how many variations in a given allele can be found in a person in a population? What is the max variation you can have? So I am a typical German Midwestern. I'm a mix of everything that ever was had genes in Europe. Everyone left a few genes in my area where I come from, Upper Rhine Valley. So I'm a typical hybrid of everything. I can safely assume that most likely on every single gene I have at least 2 different alleles. Why can I assume that? Because my parents are not known to have a lot of inbreeding, and their parents also didn't have a lot of inbreeding. So the assumption is there's a lot of mixing going all the way through. So for me, in most of my genes, I would assume I have 2 different alleles that are slightly different, one from mom and one from dad. So, 2 max you can find in a given person. How about all of us here? We all look different, and we all look different because every one of us has different alleles of the same gene. So let's see, we have about roughly 130 people in here, something like that. Let's assume about 200 different alleles for every one of our 20,000 genes. There's a whole lot of alleles floating around here. Okay? That's why every one of us is different. Because everyone is a unique mix of different kinds of alleles. Okay? So in a nutshell, basically what we are talking about here is 2 in a person, unless you are really bred highly through inbreeding to homozygosity at every single allele, every single genes, and we're talking about hundreds in the population. Remember when we started out on our Punnett squares? So do a Mendel-Punnett square again. To do a Mendel-Punnett square, we can easily answer that part. Most of you don't probably even need to do a Punnett square. Can I, if I would be a bleeder, inherit my X gene to my son? What are the chances? 0. Okay? Because I get my X to my daughter and my Y to my son. That's why my son has my Y. Otherwise it wouldn't be my son, it would be my daughter. So now the second part. What does mom do with that? Well, mom is the culprit for my son's bleeder. Because she has one allele, it may be a carrier. But because the other one is normal, she's recessive. He doesn't know. So what is then the chance for my son? Depending on the genetic background of my wife, would depend on whether she is carrier or not. If mom is carrier, 50-50. Right? So either one or the other X chromosome. Are we all with me again? It's going back to the very first hour. A little bit of genetics. Read the question and think about it. There's only one possible answer. The gene has 3 alleles, 2 are codominant, 1 is homozygotic recessive. In this case, you have a father who has the 2 codominant alleles and the wife who has the homozygotic recessive. What are the chances that the child will carry both codominant alleles of that couple? Remember, recessive, 2 alleles are then, in order to get that phenotype, codominance, you have the 2 alleles A and B. Now you cross the 2 alleles A and B with the 2 recessive alleles. You cannot get A and B out of it. You either can get one of the codominant or the other of the codominant co-localized with a recessive allele. The case we talked about was the ABO system, where we had the AB blood group and the O blood group, and really it doesn't work that way. You either would have A or B, but not AB in the children. So the chances of getting something out of it were in a cross between one parent being homozygotic recessive for a gene and the other one has homozygotic codominance is 0. Moving on into yet another area. Thinking about one of those parts where the book went through, and he looked at a number of logical steps taken to evolve multicellular organisms. What are minimal essential requirements for a single cell organism to turn into a multicellular organism? We talked about this at length, one of which was that one. You made a contact. The other one is what I'm currently doing. You talk to each other. So you need to develop a language where you can talk to each other, the chemical language, and you need to have the ability to touch each other and make contact and stick together. If you don't stick together, you just float apart, like you are leaving that room eventually. Okay? So that's minimal essential requirements. You need to have those properties. And we talked about the molecular basis of it. All right, I know this is already 3 weeks back, but we bring it now back up to the upper level of your consciousness. We went through evolution. We had a number of arguments why evolution is pretty much the only explanation in town that fits all the facts. One of the arguments was, well, if we take embryos of different vertebrates and compare them at similar stages, they have a high level of resemblance. And then we discussed what happens with these embryonic structures as we move forward. Really, in a nutshell, you are recapitulating to a certain extent evolution, because you start out your life with a single cell. And you become a multicellular organism that is really shaped like a ball of cells and eventually undergo all the differentiation. But what I have in mind here in particular is this case. The gill arches that you find in fish that have gills attached to it, we developed the gill arches as an Anlage, but we turned them into your lower jaw, your hyoid bone. And what we did is we touched our larynx, and we all swallowed. We felt the darn thing is moving up because the hyoid bone is yanked up when you swallow. So we all hear about how bad we are in terms of mass extinction to our environment. We're killing more species so that the boulevard was healthier than anybody before. Unfortunately, it is not quite right. What was the biggest mass extinction we know about, and who caused it? The dinosaurs, right? No, not really. What do you think? Complete wipeout of an early life form that dominated that planet for maybe 500 million years. The culprit is what you use right now as you inhale. Oxygen. Okay? So remember, once upon a time, there was no oxygen on this planet. Once photosynthesis was invented, chloroplasts were invented. Photosynthesis started, and as a byproduct, a waste product of photosynthesis, plants secrete oxygen. Before there was no oxygen, your life would be very short-lived in the old earth. You had no oxygen to breathe. So that revolution of making chloroplasts, making photosynthesis, and generating oxygen as a waste product generated a complete new world out of which we evolved. So that was probably the biggest mass extinction, and we don't even know how the critters looked like, who were all killed, because that was all single-celled organisms. back to genetics. The things you shouldn't do, rather insist on marriage, because then you are turning into what is called isogenic. In other words, the two alleles we talked about that you get from mom and dad, all by a sudden are the same alleles. So what is then happening to you when you want to make gametes out of it? What variation would you have on these gametes? How many different kind of gametes with respect to that gene and those two isogenic alleles can you make? What degree of variation would you think you can produce? 20, 10, 5, 2, 1. You're isogenic. You have no variation anymore. So there's nothing what you can vary. It's one and the same allele on the two loci of your gene. Consequently, it will be the same on all of your gametes. If you suspect a trisomy, what would you do? What would be the easiest way to identify a trisomy? Karyotype. There we go, the karyotype. You look at the karyotype. Okay. Maybe very simple a technique, but it still is out there and it has its efficiency, because you simply see, this chromosome comes in 3. That's why it's called a trisomy. Okay? Easy. A little bit more complicated. How do we correlate the phenotype and the genotype? So most of what you are is your genotype, translating itself into phenotype. But if I start you when you grow up, what will happen to your phenotype? Yeah, so we need to have, the king need to work on that part, namely the most need, right? Okay. Because there is an influence of your environment on how your genotype is translating itself into your phenotype. If I would have grown up in Africa by now, I would be very tainted in my face and likely would already have a skin cancer. But that would be a problem because I don't have enough melanin in my skin. Okay, so that's where we have to think about our environment. Remember, the female meiosis has the strange phase where you do mitosis 1, where you form a polar body and the egg with the nucleus, and then only when the sperm enters the egg, you are now undergoing the second division, where you then reduce the total number of your chromosomes down to the half set, so you can fuse it with your sperm. So now you think about, could you actually have a sperm that fertilizes the polar body, that little thing that carries nothing else but part of the female nucleus? And what could you get out of it? A, it doesn't happen. B, if nature would really play a joke on you and let it happen, what would be the consequence out of it? Remember, your egg is undergoing the cell division as it travels through the entire oviduct down to the uterus. In order to do so, it needs to be supported by the egg yolk that is in the egg. The polar body has nothing. It would not even last long enough to undergo a few divisions. So even if there would by accident anything happening along that line, you would not, nothing would come out of it. Okay? Why do sperms have nitrosomes? So we can find a new way to name something really strange, and you have to learn that word. What would happen if a sperm has no nitrosome? It contains enzymes, and the enzymes are needed to penetrate into the egg. If you don't have nitrosome, the stand next to the egg and say, sorry, I'm here, but you have to open the door for me to let me in. So the acrosome is literally the key to get into the egg. Sperms without acrosome don't go to the next generation with their genetic material. Okay? Literally, you digest your way into the egg. We talked about the three-layered embryo, with the endoderm, and we talked about the endoderm is your guts and all the major glands on your guts. We talked about the mesoderm, that is everything in between. And we talked about the epidermis that gives rise to the ectoderm. And then something else. What was the something else? Do you have it? Anyone thinks that something else is not there by that, in their case? Because you would have a real problem. You wouldn't be here. It's called the brain. And I had once upon a time on my office a note of all the things I lost. I miss my brain the most. Right? So you don't want to miss your brain. They're not very funny. You're supposed to laugh about that part. Okay? So epidermis and brain are really coming together. What are we? One of a kind. You cannot be compared with anybody else. The concept of taxonomy has been changed over the last 100 years. 1920, a famous taxonomist coined the word. His name was Reagan, not the Ronald Reagan of presidency, but a guy who actually understood what he was doing to a certain extent. So this Reagan said, a species is what the competent taxonomy said it is. What he was concerned about is taking animals and put them together in certain categories, right? So one of his saying was, species is what a competent taxonomist says it is. And he basically also said, well, human is really a big ape. Okay? So the taxonomic composition of humans can actually be written in a single line like this. We belong to the primates. The primates are part of the mammals. Mammals belong to the tetrapods, and tetrapods belong to the vertebrates. Okay. So we belong into this entire group of animals that are called vertebrates, the guys with the backbone, as compared to insects who don't have a backbone. Okay. And you should have a backbone, hopefully. What is meant with genetic drift? If you sit in your car and you drive from here to Kansas City, is that genetic drift? No. Because most likely the people living in Kansas City and the people living in Iowa are the same ethnic mix, so you wouldn't add much to the genetic mix that is already out there. So what do you think when we talk about genetic drift? Founder population, an island, a few people living in there, and then start to propagate a new breed, right? That's what we think about when we talk about genetic drift. A random isolation of a small population through a catastrophic event. For example, the continent are ripping apart, and a few isolated species happen to be on Madagascar. Since they can't pop against back into Africa, they evolved into their own line, and they became the lemus. So lemus as a group is only in Madagascar. That's the background for that. That's one of the little bit more complicated components I want you to think about it. We all use it. Look at somebody and say, he belongs to that group, he belongs to that group. But really, we cannot do that so simple. Because what I try to get across to you is the color intensity of your skin is dependent on one single variable only. The amount of activity of a single enzyme. So there you are, you have 20,000 genes. And now you want to make a conclusion about the function of somebody based on the activity of a single gene that in addition is regulated by sunlight. Because if I go up to the North Pole, I turn really pale. If I would live in the equator, I would be a little bit more colorful. So the bottom line is, in common terms, we are taking a fraction, a teeny little amount of the total number of genes to expand it to give some kind of an information content to a specific colored group of people. The blonde guys, you all know the blonde jokes, right? Well, it is not true, guys. Not all women who have blonde hair are really a little bit on the cortically challenged side. And so it goes. But we all do this every single time, taking one single obvious character to cast one group of people in a specific light, forgetting that we have another 19,000 plus genes. Assume it's not late in the party. Assume you are still there and your cortex works and you have not so much alcohol that whatever you talk about is meaningless. What would you say? There was this guy who told us so. That's not good. That's not a good argument. An argument is logic. What is the logical argument you would come up with? Carbon dating is a good one. So you dig down in a big rock, and 2 feet down below the surface of the rock you find a fossil. The fossil is also rock. It's bone turned into rock. What do you think how long it takes before your bone, when you die, turns into rock? 2 weeks? 5 years? Think twice about it. We are talking about thousands of years. Okay? So here's the argument. You simply turn around and say, well, wait a second. It takes a little while before bone turns into stone. And besides, we have radiocarbon dating. And radiocarbon dating is essentially a physical principle that cannot be changed by anyone.